I can't admit people if you want me to help,
Oh,
because
I started record. It's fine. I just
okay,
started recording.
all right. And in terms of admitting people, so can you do that? Or but it would be
Uh,
difficult. But if you need to give me some kind of co-host so I can
yeah, I
admit
gave you
people.
co-host, I think. So I think you can also do it. Maybe it's just a little bit less convenient on the phone. But
That's why I'm trying to figure out where. Uh, more. Blah, blah, blah. In Jordan today starts. There's the higher education summit.
okay.
THE that ranks the universities. So the Arab Summit is in Jordan tonight and starts tonight, and I'm one of the speakers.
Oh, wow.
So and so we need to start gathering ourselves at 6:30. So I'm gonna be on and listening, but I'll have my earphones, and that's why I couldn't do it on my laptop because I'm not at home.
Okay.
So and I don't know where to figure out how to admit people. Let me see. Yeah, I think the phone has limited. And are they entering or are you entering them?
I'm entering them right now.
You're
I can see
admitting
them.
them. So is that that's going to be inconvenient? I know. Sorry. Yeah, we're multitaskers, aren't we?
Yeah.
Okay. So I will begin, and we'll make it happen. So welcome everybody. Very nice to have you all, students, professors, faculty, anyone who's interested in biology. You're all welcome. On behalf of the Committee for Culture at the Department of Biology and Biotechnology, Faculty of Science at the Hashemite University, we are very excited to welcome today Dawn Chen. Dawn Chen is a PhD candidate in systems, synthetic, and quantitative biology at Harvard University. Her research focuses on developing new genetic engineering tools to understand how genetic variation affects cells and how to precisely control gene expression in specific cell types. Before Harvard, Dawn graduated from Yale University with degrees in molecular biophysics and biochemistry and statistics and data science, where she discovered new RNA control elements and studied genetic risk for cardiovascular disease. She then worked at Google as a software engineer. Born in China, raised in Singapore, Dawn is passionate about making genetic testing and precision medicine more accessible around the world. In her free time, she enjoys hiking, experimenting with new recipes, and exploring unusual musical instruments, which we need to get to the bottom of that. We haven't.
We need to
Um,
talk about that another
yeah,
time. Yeah.
I got to know Dawn recently through a project working on ethics and data sharing and data visitation and localization of data, which is very exciting. And we're working on a number of other projects as well. Today, Dawn, please mute yourselves. معلش تسكروا الميكروفونز. Today, our lecture will be about AI for biology, which is a very, very hot topic. It's all groundbreaking. And as Don said, it's changing every day as we speak. So we're very excited to learn from Dawn. And then, Dawn will speak for around 40 minutes, 45 minutes, and then we'll open it to Q&A. You can write your questions in the chat, or you can just unmute yourself and ask the question. اللي بده يحكي بالعربي أهلا وسهلا بنترجم له مشكلة، أو يكتب في العربي وبنترجم له بالشات. هاي فرصة نتعلم من بعضنا البعض ونستفيد من بعضنا البعض. فأهلا وسهلا بالجميع، ونحن عم نسجله مشان كمان آخرين ما قدروا يحضروا يستفيدوا إن شاء الله. Without further ado, Dawn, the floor is
Okay, I can
yours.
get started. Yeah, hello everybody. I'm so excited to be able to give you this talk today on AI in biology. And I think actually I'm not really that qualified to give this talk just because there's so much going on in the world of machine learning and computational biology that there's just like new things coming out every single day. And every day I'm learning new things. But I would say that in my own personal work, I do spend a lot of time building models. And also, I used to work in software engineering. So, this is still a topic that I guess I know a little bit about. And I guess today I'm going to give you more of just broad overview of what are some of the applications of AI right now. Maybe not so much go into the technical details because I think they can get really complicated very quickly. Um, and I think I was just start out by saying that, biology is highly, highly, highly complex. On this slide, you can see this is a map of all the different biological and metabolic processes that are in a cell. And this is just a chart. And you can see that there's just like so many nodes, so many different metabolic pathways, so many different parts of the cell. And we really need some way to make sense of all the complexity of data that we have. And what is like artificial intelligence in biology? Like, everyone knows that AI is this like super hot term today. But I would say that like at its core, really, AI is really a way or a set of algorithms so that we can understand, predict, and manipulate biological systems using. Because there's just so much large data sets out there that we need some way of being able to analyze these large and complex data sets. And some of the things that AI really is that at its core, it's really about a lot of math and statistics and data analysis. Like maybe you have like plotted a lot of graphs before, yeah, like analyzing people's height versus weight correlations and things like that. On a more like advanced level, there are also a lot of is also machine learning models. For example, random forest, support vector machines, logistic regressions. And these days, there are also more like deep learning models. For example, like neural networks, transformers, foundation models, which are terms that you might have heard about. And newer than that is there is also now a lot of generative AI, or for example, using AI to design new proteins, sequences, or small molecules. Or maybe you might be familiar with like ChatGPT is a form of generative AI. Like you give it a prompt, and it can just help you, and it can just be able to generate new content for you. And there is also this emerging paradigm of AI scientists, which is AI agents that can help you plan experiments and maybe even execute experiments so that you don't have to do a lot of the thinking because the computer is smart enough that you can actually use it to help you automate a lot of things. And I think the next question is why might we want to model biology or why might we want to ask a computer to help us understand biology? I think there are a few reasons that people have agreed upon, or generally people will agree with, is that first, we really have lots and lots of data and we need to make sense of it all. Like we have collected so much single cell data, we have a lot of DNA sequencing data, we have a lot of imaging data. Like if you go to the doctor and you take a CT scan or you take an X-ray, then that's just like so much data that we need to analyze. And it's actually really hard to analyze a lot of this data. And I think the other thing that is very interesting is the reason why we want to model biology is that the language of biology, like DNA and RNA, really is a programming language, right? Like your DNA has ATGC and it's a set of instructions and makes it into RNA and then translates it into proteins. And so it follows underlying rules of patterns and structures. And so we can use modeling as a way to find the structures in the DNA. And modeling is also very useful because we can use computers to help us explore new hypotheses that might not be intuitive immediately to humans because humans are smart, but maybe we can't just observe patterns so easily across many things because we also just have limited time. And finally, if we can really predict outcomes computationally, we can definitely save time and resources by running fewer experiments. And maybe some of you have been doing that. Like if you write an essay, sometimes I use ChatGPT to help me paraphrase things so that I can make a much better essay much more quickly. And so these are some of the reasons for that. And so like AI with deep learning started a long time ago. In the 1940s and 50s, people already started talking about using artificial intelligence or this concept and deep learning. Like this is where the first like neural network perceptron was kind of invented or people came up with it. But then like it took a really long time, maybe almost 50 years before the first algorithms really started to become much more mainstream in use for research applications, but also more consumer applications. Because for a very long time, these were more of just like mathematical, theoretical problems in computer science that people were working on. And then afterwards, people started in a research field to look at all of this like data, to use this data to be able to analyze patterns, more so in the realm of machine learning, like classifying cats and dogs and more of some of these things. And then obviously these days we have reached the age where we have things like ChatGPT, DeepSeek, Anthropic, there's just like tons and tons of companies that you have these models that have much more become consumer grade products. And the reason why there's this such a shift from kind of just theoretical to apply to really consumer is I think there are three main reasons of why AI is so hot right now, especially in biology. And these are mainly due to three reasons. The first is we just suddenly have massive, massive amounts of data in biology. We have better tools for DNA, RNA sequencing. We have electronic health records and also imaging data, so that we need to analyze and make sense of. Like it's like overwhelming amount of data. The next reason why we can do AI for biology really well now is also compute power. People spent 20, 30 years like NVIDIA to work on faster GPUs that are cheap, that, people can just do a lot of analysis on very quickly. And third, obviously, is that there have been a lot of improvement in theoretical to work on these algorithms. Like, for example, people needed time to be able to come up with the math on neural networks and transformers. And next, I want to talk a little bit about the difference between predictive and generative AI. And so very commonly, people use a lot of predictive AI, which is making predictions based on inputs. So what people would do in this case is you might train some computational model with available data, and then predict features for unseen data. So this is maybe something like, given a number of shoppers who visit a supermarket today, I can predict how much money the supermarket is going to make today. And then there's generative AI, which is you can generate new contents. For example, ChatGPT can generate an essay for your class given a prompt. And I think in both cases, what is really important is we need a lot of data, because if you don't have good data to teach a computer how to learn or what to learn, then you actually can't really learn. You can't really figure out what to do. And so today I'm going to go through a series of topics of applications of AI in biology. And this is just going to be a pretty broad overview of how AI has been applied in different areas of biology. And some of this stuff is really like every single week you can find a new paper. And a lot of these things you actually can just like try out what they have made. And finally, I'll also cover a little bit about what are some of the current limitations and ethical considerations, and also how I can start learning AI in biology. Just some practical tips for if you want to get into the AI space. So the 5 topics I'm going to cover today are structural biology, like protein design, genomics and diagnostics, image analysis, drug discovery, and also the use of AI scientists for experiments, or AI for science. So the first part is about structural biology. And you guys might know that last year, the Nobel Prize in Chemistry was given for computational protein design and protein structure prediction. And these were given to 2 groups of people. The first is David Baker at University of Washington, because he made a lot of tools that enable people to design new proteins. And this was really and kind of synergistic to that is there's also other people like Demis Hassabis and John Jumper. These are people at DeepMind, at Google DeepMind, and they made things like AlphaFold that can really do protein structure prediction. And so these kind of prediction of structure and designing new proteins really come hand in hand. And I would say that protein design is really like, I would say, the hot thing right now. And so what is this thing that they did, and why is the thing that they did important? And so, there is basically this protein folding problem, which is that proteins are very structured because we know that proteins fold into very, very like good structures. And scientists always want to know the structures of those proteins. But historically, experimentally, it's actually a very tedious to figure out what is the structure of a protein. Because what you would do is you perform something like X-ray crystallography or cryo-EM. And this is like a super, super, super tedious process. For every single protein, it takes 1 month or even years to make the protein crystals and then purify them and finally get a structure and solve the structure. So wouldn't it be easier if we can computationally predict the 3D structure of proteins using just a sequence? So that, let's say if I just find in a random bacteria somewhere, I find a new protein sequence, I can just figure out what its structure is without doing any experiments at all. And that will definitely be much faster. And so AlphaFold by Google DeepMind is this thing that was this algorithm that was made for protein structure prediction. And it actually generated like so much hype around the world. And it's just like they just kind of like beat the next best method by a lot, a lot, a lot. And so like what does actually AlphaFold do? It basically takes a protein sequence and then predicts its structure. And actually, this is something you can try right now. You can actually just Google AlphaFold DeepMind, and then you can actually just go find a server. You can enter your sequence and look at a protein structure. Actually, I don't know if any of you have played around with it. It is pretty cool and it's free, so try it out. And the reason how AlphaFold can be trained is because there has already been like, over the period of like decades, people have already collected this thing, a lot of protein X-ray crystallography structures called the Protein Data Bank or PDB. So AlphaFold is trained on all this open source data from the PDB of protein structures where you have the sequence that converts it into structure. And so that's why like there is this like AlphaFold, and so like data here is so important because if we didn't have a good data set of protein structures that already existed, then there is no way that we will have be able to train a model to become something like AlphaFold, because you just need to know you just need to have some knowledge of protein folding to begin with, and obviously these people did some really cool algorithm advances such that they can be able to get this like structures a lot better. And so how do people generally use AlphaFold, or how you might use the AlphaFold is that first is you might want to predict protein structure, and this is very valuable when there is no experimental structures available, because it's just very, as I said, very tedious to generate experimental crystals. Next, you can use it to help annotate proteins, like for example active sites or ligand binding, and also help to model protein-protein interactions, and also lead to how different mutations lead to disease and so on. And so after you can model structure of proteins, what else can you do? The next thing that you can do is you can do de novo protein design, which is how do you create proteins from scratch that don't occur in nature. And this is really like cool, right? Because like we now know so much about biology that we can not only understand biology but also engineer biology to do new things that never existed before in nature. And that's what is the beauty of de novo protein design. Like for example, if you want to have a new antibody, like these days maybe you have to do a lot of experiments to get it, but what if I just think about this idea in my head of like, okay, I just want a new drug, a protein drug that it should have these properties, and I just tell the computer that, and then the computer just generates something that just works for you. Like that's kind of like the epitome of like what computers should be able to do of generated design. And so what are some things that de novo protein design can do is that some of the things it can do is create new protein shapes, design new protein binders and antibodies, make better enzymes, develop new therapeutics and biosensors and so on. And the limits are endless. I will also say that this is still actually very much an emerging field where like, like I would say currently the hit rate is about maybe like around 5% to 10%, where that means that if you test like 10 designs, usually one will actually work well. But this is still like a much better method than what already exists before. And so if this is an area of research that you want to work on and explore, I highly recommend you like looking into it. And actually, I also have at the end, I will give you a set of references that you can actually look at to learn more about it. Yeah, and as I mentioned, you can actually use AlphaFold these days to design your own proteins. And so this is actually like an AlphaFold server uh, that is available. You just go play around with it. This is like by Google. And Minecraft is the state of the art for protein design. Is like this other like newer tool that someone made for designing binders specifically. It kind of integrates some of the protein design algorithms from David Baker and also AlphaFold to be able to like kind of figure out the structures of proteins. Okay, so that's protein design. Next is about genomics and diagnostics. And so here, what is the goal, right? Like our DNA contains 3 billion bases, and one of the fundamental challenge of biology really is to understand the role of every single DNA base. And this is really important for the case of genetic disease because maybe we just want to know, which of this DNA sequences and changes in DNA actually leads to a disease. And so generally how people have done this is they have done a lot of observational study, right? Or what you call GWAS, so genome-wide association studies. And what you do in that case is you take a lot of healthy individuals, you take some patients with disease, you kind of maybe sequence both populations, and then look at how their DNA is different between the healthy people and the diseased people. And by that, you can then figure out what are some of the mutations or genetic variants that are associated with the disease. But obviously, that is like correlation, not causation, because at that point, you actually haven't proven like which mutations lead to disease. And then the other way that people have been studying genetic variants or figure out how mutations lead to disease is, of course, through running experiments. So, for example, what you can do is you can take a dish of cells and then you can introduce individually by things like genetic engineering, which is like techniques I talked about last class, to introduce these mutations to cells at scale, and then you can do some sort of functional assay, like look at which cells survive or which cells turn green or something, and then read out the cellular phenotypes, which will tell you then like which of these genetic mutations are important in causing disease. But both of these actually are like really tedious processes because let's say you do some sort of observational study. You go out and you find like maybe you can find 1,000 patients, a few thousand patients at best, or the biobanks, maybe a few million patients. But you still have to like pay them a lot of money, get them to come, and it's just like really hard to collect all the data. The same actually goes for experimental study, which is that, this is very hard work to be able to do these experiments in a lab. So what is better is what if you can just use like computers to be able to predict the function of every single DNA base, where let's say you have just a series of DNA and then you can put it through some sort of machine learning or AI model, and then it can just tell you what each single mutation do and what is the effect size, right? And people like Alpha Genome from Google actually did that. And there are a lot of other models that have done it. And this actually matters a lot for research and clinical diagnostics. For example, just think about, let's say a sick patient comes to the clinic and then we think they have a genetic disease. We sequence that patient's DNA and we found that they have a lot of mutations because it's very common if you just sequence someone, you will see that they have maybe on the order of 500,000 different genetic variants. And not all of them obviously is the reason that causing them to be sick. So then how can we know which mutations actually lead to disease? Obviously, one way is we can test all of these individual mutations individually, but that takes like so long. It will be so much better if you can just like predict the effects of these things. And so that's what a lot of these algorithms actually try to do. You have things like Evo2, Alpha Genome from Google, DeepMind, Deep Variant. This is AlphaMeSense also from Google, and this is like Illumina Splice AI. And what all of these do is it actually like you can give it a genetic sequence and a genetic mutation for the most part, and then it can tell you whether a mutation leads to let's say a change in protein structure or a change in alternative splicing. And this is like a model way of like how we can make this just use computationally to be able to predict the effects of different genetic variants. And I would say that like all of these methods work reasonably well, but there are also obviously problems, right? As I mentioned before, in a protein case, you can only learn what you already have data for and what you already know. So we only have so much experimental data that's been generated for genetics and DNA, and it doesn't cover every single part of the DNA. So obviously, we still need to develop better, just collect more data so we can train better models, so that we can really go to be able to understand all of the roles of every single mutation. And then the third part I'm going to talk about is the area of image analysis. And image analysis is very, very common for things like radiology and pathology. So maybe you have gone and get an MRI before, or maybe, if you have family members, unfortunately, who have cancer, you might have seen them get a biopsy where they will look at tissue sections. And then so how people do it is that say this is actually a tissue section of someone with cancer. And and then and then you can see that okay, you can very well tell that actually this is like, so the dark purple actually is nucleus. So then this is a section with a lot of cells and a lot of nucleus. So this is like cancerous tissue. And obviously what happens now is you might have some of these images, and a person, a doctor, will manually go through and look through all of these images to figure out, okay, what is the disease that this person has, and how can we classify them, is what stage of cancer, or what problems they have. But this is like very tedious, and obviously human eyes can miss a lot of things. This is like playing spot the difference every day. And so what you can then do is, obviously you can train AI models, because we have so many thousands of slides of this MRI and tissue slides, that we can use it to detect subtle patterns that human eyes might miss. And obviously computers can also analyze images faster than humans. And so AI really can be another pair of eyes, like for example in computer vision. And one of the things that it can do is this thing that's called cell segmentation. So the first thing that you will do is, this is an image of a lot of cells. The first is you can actually like segment out the cells to figure out where each cells are located. And then you can kind of think of making a set of algorithms to be able to analyze, oh, is this cell cancerous? Is this not? And so on. And this is what there are a lot of like packages that you can actually also play around with, that you can use, like Cell Profiler, Cell Pearls, Deep Cell, StarDist. And actually, I highly recommend, if you're interested, you can go and play around with some of these tutorials. For example, Cell Profiler is actually, it has a software that you can just download, and it can teach you how to do cell image analysis. And it has a lot of tutorials that you can walk through to learn how to do image analysis. And so what are some of the applications of image analysis in biology or in the clinical world? Is that you can use it to detect stroke or hemorrhage from CT scans, detect breast cancer from mammograms, because sometimes the doctor may look at an image and be like, hey, I don't think there's cancer, there's nothing on here, but maybe if the computer can just look at it much more, scan it a lot more with much more detail, then you might be able to like miss things that you might find things that doctors might have missed. You can screen people for lung cancer, detection of like cancer metastasis, or sorting of biomarkers. Basically, anything that you can do as a human that your eyes can see, you can more or less train a computer or train an AI model to be able to do that. For example, in this paper, this people made a model that can classify anomalies in chest X-rays. So here you can see different people with different X-rays of the lung, and then you can classify things of like, okay, this is a mass, this is a nodule, this is pneumonia, because they look a little bit different. And this is also possible because there are a lot of images that are already available, data that people can train their AI models on. So then the next part of it is drug discovery, and so AI actually really is accelerating drug discovery, and this is a part that I would say a lot of pharma companies are very interested in, because like drug discovery is a very, very expensive and very long process. So if you can just speed up the drug discovery process in any way, that will just be so much better. And here's a chart or a summary from this Nature Reviews article of how AI has been used to identify new drugs for patients. And so if you were to just think about a pipeline of drug discovery, what people would do is first you identify a new target. Let's say KRAS is associated with cancer. This is like a very well-known target. Now we need to figure out how to target this protein that's called KRAS. Then afterwards, in the case of small molecules, you then have to figure out, okay, what are some of these small molecules are active compounds or lead compounds, which are things that will bind to KRAS and knock it out or something. And after you figure out what is the best like chemical small molecule, then you can do some sort of preclinical studies. You put it in animals, you try to figure out that, okay, this molecule that I found actually is doing what I want it to do. And once you have done that, then you can do clinical trials in humans, where then it's like, okay, I give a patient a drug, I can see how the patient performs, does it cure that cancer, and so on. And finally, after you do clinical trials in humans, then finally you can approve this new drug and say, yeah, I now have a new drug that people can use. And then even after having a new drug, you might want to do some sort of post-market surveillance or just monitoring of how people perform on a drug. Like what is its efficacy? Are there any side effects with people? Like do people actually respond to this drug? And so this pipeline really is a very long process. I think from target identification to approval for every drug, this generally takes up to 10 years or even more than 10 years. And a lot of drugs fail. So anything that can accelerate this process is very helpful. And so how AI has been used in a lot of this analysis is first, maybe you can do some sort of literature mining to figure out, okay, what are actually some of the new targets we can target. Then the next set is generally people would just like discover these active compounds by actually doing experiments of putting different chemicals onto cells to figure out what is the problem with the cell, which of the drugs actually inhibit the cell. But this is a very tedious process. And so there is this new idea called virtual screening, which is I just screen compounds virtually on the computer. And so then you can actually screen many, many, many compounds very quickly. And then after this, you can then also use AI for different use cases that I'm not going to like kind of go into in detail here. And so, as I mentioned, AI, a lot of these AI tools have accelerated drug discovery pipeline in some ways. For example, instead of performing experiments, people can now perform virtual screening to be able to identify promising chemicals using computational methods. And one of the example companies that's working in this space is a company that's called In Silico Medicine. And In Silico Medicine was able to get a few of these drug targets in the clinical trials a lot more quickly than standard, because they were able to screen many, many, many compounds very quickly, and so they saved them a lot of time, and obviously that means they saved them a lot of money. I don't really know. I think they're still in clinical trial, or I don't really know what happened to them yet. So it's still a very rising field. But I will say that if you were to just look around, there are like tons and tons of companies who are working on using AI tools to accelerate drug discovery in this way. And I also want to mention this paper, which really was a pretty landmark paper that came out in 2020, where researchers at MIT, actually these are the people who sit beside me in the room next door to me, and they used AI to develop or discover the first antibiotic that's called halicin. And so antibiotic resistance is a very big problem, and we need to find new antibiotics. And so here what they did is the MIT researchers trained a deep learning model to predict whether individual molecules can inhibit bacterial growth. And so they just give the computer a molecule and then ask it, okay, does this kill the bacteria? And then get a set of metrics. And using this process, they were able to screen more than 100 million individual chemicals in a span of days, instead of the many months or years it would have taken them to actually run those experiments. And also a lot of these molecules, like chemicals, you can't just buy it randomly from a store because it doesn't yet exist, right? So if we can screen things, computationally just be better. And so this is the structure of halicin, which is an antibiotic that it was discovered. Actually, this is already an approved drug. So then they are trying to, I think, do some trials to make it work, but people have already been using it for other indications for a while. I think in diabetes, if I'm not wrong. So they found that this drug, which was used in diabetes, actually is also an antibiotic. I might be wrong on the diabetes part. It was some other disease. Okay, in the last part, I kind of want to talk about what is like the next like really hot thing right now, which is like AI scientists. Just like really briefly. And what is an AI scientist is that it is a virtual scientist that can generate new hypotheses, analyze complex data, and design new experiments. And they can also be integrated with lab robots to be able to run experiments directly. And most of these, what these are, is that they kind of are just a wrapper over ChatGPT, you can think of it, because they just kind of integrate ChatGPT with a little bit more computational biology tools to accelerate your research. And The good thing about a lot of these tools is that you can actually try them and they're free. So, for example, I don't know if you've heard of Biomni, which is a general purpose biomedical AI agent that was developed by someone at Stanford. And then there's also this other Future House platform, which is also trying to build AI agents for scientific discovery. I would just like kind of look at their articles and read through them, and also just play around with them. And these are just the.
Oh,
Mhm.
yes, what's the question? I asked people to think so. I'm not an associate professor. Did they think so? Okay.
Hey Rana, am I gonna? Can I mute you?
Good. Is there any particular scene?
Okay, I just muted Rana. I think it's fine. We can continue. Yeah. Um, Finally, I'm going to talk a little bit about the limitations and ethical considerations and what is AI good for and what is it not good for. I will say that AI models, even though there is a lot of hype today, there are still a lot of problems because I think on the surface it looks like okay, AI is going to change the world. I think it's going to make the world a lot better place. But there are still a lot of limitations that we should be aware of, which is that first there is still not enough data. As I mentioned, AI models really need to be trained on loads and loads and loads of data. And for many areas of biology, we still don't have enough data and just need to collect more data. There is also a lot of bias in data where many of the data has only been collected from specific groups. For example, genomic data is largely collected from white European populations, and this can exacerbate health equity and also lead to incorrect predictions. And finally, many AI models are black box, so they are not really explainable. We don't really understand what it is specifically that these AI models are learning, or is it learning anything that is biologically significant? And so we need to validate them before using their outputs for clinical decision making. Like for example, if you were to give some a patient's DNA and then it tells you, okay, XYZ mutations means this person is more likely to get cancer. How likely are you to trust it that it is actually true, or is it just the AI just making stuff up? So we still need to be able to understand its thought process to be able to have better models that are much more trustworthy. I think on the other side, there's obviously still a lot of ethical considerations as we start thinking more about AI as something that's not just in the research realm anymore, but more widely used for everyone in the world. And maybe there are three ones that I think are be very important questions to think about. There are obviously like a lot more. These are just some primers for you to start thinking about these problems. The first is data privacy, which is that many models in biology are trained on sensitive private data, like for example genomic data. How can we benefit from these models while at the same time still protecting individual privacy? There is also a dual use and biosecurity risk, which is that for all technology that is good, they can also be misused. Like even for things like cell phone, like cell phones are very good for communication, but they can also use in the case of like defense and military, and so AI models in the same way can also be misused to design harmful agents, for example. And how can we control and monitor how people use these models? And finally, there's also a lot of problems on consent and data ownership. For example, patients may not understand how their data may be used to train these AI models. And also the people who provide the data may not be the one who is getting the benefits of this data. So how can we ensure informed and meaningful consent and data owners getting meaningful benefits? In the realm of, for example, human genetics, you might have heard of, say, like 23andMe getting into a lot of trouble because people like you kind of did this test for DNA sequencing, but then they might have taken your data and they are using your data for other purposes, like doing research on different questions you're not aware of. And so obviously we need to have more just transparency and meaningful consent and benefits from everybody. And and this is actually a question that I've been thinking a lot about and also work on, and also actually I've been working with Dr. Rana on some pilot projects on this, which is that in the world of genetic or just genomic data policy, that there is this new concept that is called data visitation and federated analysis. And what that really means is that instead of moving all the data together and analyzing them centrally, data owners can then make their data available for remote analysis without uploading or exploring the raw data. So here, instead of moving a data to the software that you're running something, now you kind of are moving the software to the question. Because what happens is, let's say I want to analyze your data. What I will do now is I'll ask you to give me a copy of your data, and then I'll analyze it. But once you give me a copy of your data, then I have control of your data and you lose the control, and you don't have control over what I do with your data. And so that's not great. So ideally, you will be the one who will have full control of your data, while I can just send code to your data to analyze your data, and then you give me back the results. And so this actually allows collaboration without giving away a copy. And we have actually been building a platform that's called BioVault to do this. It's an open source free project, and we are kind of just running it as a group of volunteers right now. And so next is finally, like, there are a lot of exciting things in the AI world, and like things are really, really changing every single day. So what are some of the ways of learning AI and building your own models? I would say that there are a few things that you can do, which is at first, you probably want to have a very good understanding of math, stats, and machine learning. And there are really a lot of online classes there. For example, this deeplearning.ai, which is a class by Andrew Ng. There's also this Manolis Callis machine learning and computational biology course. If you want to look at more like cutting edge stuff like protein design, there's also just tons and tons of paper archives that you can read. And then the other very important thing is learning by doing, because to learn how to code, how to do math, how to do statistics, really is that you just need to work on your own side projects. And there are some things that you can do for that. For example, joining the Kaggle competitions. There are a lot of them for biology data sets, but also non-biology data sets. There are also a lot of in challenges are popping up around the world. Like, for example, our institute just had this virtual cell challenge for predicting something about virtual cell. And there's this huge Discord community where people, like random people just like posting is a good way of meeting new people. And then finally, the best way to learn is just to reproduce things from papers that you read. Like, for example, you might read a research paper, and then you can just kind of go and find our code. And there are also a lot of tutorials where people have done like reproducing of their data. And once you understand how people plot their graphs, even from the simplest graphs to like a little bit more complicated model building, if you can understand how they did their thing, then you can then use it to build your own models, and so on too. Okay, that's about it. And we have some little bit of time for questions. So let me know if you have any questions. And I'll also put this slide deck actually in the chat right now. And if you have any questions, just feel free to message me. And I'm happy to answer any of your questions.
Thank you, Dr. Dawn. I actually have two questions.
Yeah.
The first one, how much accuracy does the AI tools give as for DNA and proteins models? And the second question, you can answer it. I can ask the second question later.
Oh, so accuracy of AI and protein models. I think accuracy and precision is a very important question, and actually is something that's really hard to evaluate. I think actually that depends on what your benchmark is. So for example, yeah, I think it really depends on your metric. So I would say, like, for example, as I mentioned, in terms of designing new proteins, depending on the task, which is like, say, designing a de novo protein that binds to a target.
Hello, sorry, Dawn. Thank you very, very much. Can you hear me?
Yeah, I can hear you.
Yeah, thank you very, very much. Sorry for because like I said, we have the summit that's happening at the same time. Now the floor is open for anybody who has questions.
Yeah, I think Malak asked a question.
They did. Okay, so I'm going to leave you, Dawn, to manage the questions since I'm coming in and out.
So no worries. I think we're fine. We can just have a chat.
Okay, great.
Yeah, no worries at all. Yeah, so to continue on questions, it really depends on the kind of task that you want to try to do. So, for example, in the case of design new novel protein, maybe the hit rate is about like 1 to 5%, which means that if you design 100 new proteins on a computer, maybe 1 to 5 of them will actually be correct. And maybe that kind of hit rate is like, oh, you think that's like super super low, but that's okay because in you're comparing to, like, you need to think about what you're comparing to, right? Like for example, in the case of experiments, you're screening millions protein before you get even one good hit. So that's actually a very good target rate. And then in the case of maybe say something like predicting the accuracy of whether a mutation leads to a change in gene expression, I think a lot of models actually have pretty good prediction, like 70 to 80% accuracy, and so that can be pretty good. But then it also depends on what you're comparing against and what you're missing, right? Because maybe you actually really want to get the 20% that is missing, and if sometimes in the case of disease, maybe like false negative is kind of not good either, because if let's say there's a positive data point, you might actually want to be able to find all the positive data points, and so if you're missing something, that's bad. So then there's a trade-off between false positive rate and false negative rate, and like all the stuff. So I think that's actually a really hard question, and I think it is like also very context specific. What is your second
Oh,
question?
that's so great. Yeah. My second question is, would they use the AI tool to as an innovative tool for preparing or the specialized medicine or personalized medicine?
Uh-huh.
Yeah, would they use AI for that?
Mm, Yeah, I think personalized medicine is also a very big topic, and there are many ways that you can use AI in around personalized medicine. For example, in the case of what I mentioned, genetics, you can kind of use it to find different diseases for individual people. And you can also think of it as you can maybe use AI to find these individual mutations, like for like let's say if someone has cancer, you can maybe figure out what kind of cancer they have by looking at their images and so on. And you can also kind of use it to do personalized drug discovery. Because let's say if someone has a disease that there's no drug for yet, these days actually there is a lot of talk about N of one therapies, which is like you only like one person in the world have this mutation or have this disease, and then how can we develop this like N of one therapy to treat this disease. So I would also say that AI is really a tool that can really help accelerate all parts of the process. It's not really like just like one thing that can just like solve everything. It also really needs a lot of domain expertise, like expertise in actual biology, in clinical medicine, to be able to work synergistically together to like make something. It's a little bit like a car, right? Like a car is a very good tool for you to be able to drive to different places. But then you also kind of still need to know where to drive to, because if you just have a car that goes very fast and room room, it doesn't really do anything if it's just sitting in
Yeah,
your
you have
house.
to have a destination.
Yeah. Yeah.
Thank you so much.
Does anyone ask them any questions? You can feel free to
They
ask
can
me
like
anything. Yeah.
they can detect a perfect medicine for someone based on their genomes or
Um,
genomic data.
there's this company or this nonprofit called Nlorum. You can look it up. They're actually developing N of one therapies for patients.
Can you
Where
write the name of the company?
here I
Oh,
sent you
yeah.
a link.
Yeah. Thank
So
you.
because there are a lot of individual people who just have genetic diseases that are very very rare.
Yeah.
You need therapies for them. So how can you do N of one therapies for people?
Yeah. Very hot topic. Really. Thank you so much.
Yeah, you can ask me any question. It can be about the talk, about not the talk, anything.
Yes,
Yeah.
thank you, Dr. John. Yes, and actually, I was wondering about it since a lot of time. In this moment, there are a lot of AI tools that are available for general use, like ChatGPT or Gemini or other that tools that anyone can use it. How can we customize this AI tools to support us in our field, like training it, or actually it sometimes it um yeah, I think my idea uh, supposed to be clear now. I'm sorry.
Oh, like how can you use these AI tools to, for example, help you with the projects you're working on?
Actually, no. I asking about training AI tools um that general, not specific to one field.
Or like how can we train these AI tools? I think the biggest part is just we just need to have better data and to train it on more data, right? Like cause like right now ChatGPT is really good because it's trained on the entirety of the internet and Gemini too. All the internet, Reddit, all the newspaper articles, all the research articles, which is why it is able to understand everything. And I think in a lot of, which is why it can only give you what it knows, right? Like it does things that in a lot of bias ways. Say if you ask it, okay, generate image of a CEO in a company, it more or less will generate someone who is white and male wearing a suit because that's just like what people learned. Um, but then ideally, if you have just like more training data, then you can kind of fight against a lot of these biases. And I think that a lot of people are also working on training AI models from using proprietary or private data that only they collect. So like the BioMe thing that I mentioned to you. Actually, I highly recommend you just check it out and play around with it because they basically made a generalized like an AI tool for biology. It basically is like ChatGPT, but then they also integrated a lot of individual just tools that only biologists would use because ChatGPT is just like very general purpose thing. But then you kind of need to make an AI agent that's like very specific to every single industry. So Biomni is one example for biology, and then there is Open Evidence, which is a name of a company that makes AI like ChatGPT for the medical field. So you can also check out this link. And here is that you can ask it something very specific to the medical, like a medical question, and then it can answer to you like what drugs should I use and what dosage and so on, and Open Evidence will know it. Versus you can actually try asking ChatGPT the same question and Open Evidence the same question and compare and contrast the results that you get. One of them is going to be a lot more specific, but these
Yes,
are
yes,
domain
that
expertise.
happens.
Yeah, just play around with it.
Yes,
There are a
thank
lot
you.
of these tools out there. A lot of them are just kind of like wrappers of ChatGPT plus something else. Yeah, at its core, Open Evidence is also using one of these like LM models at its bottom.
Yes, thank you. Actually, I have another question.
Uh huh.
Uh, it's about, you mentioned that issue, black box issue. How
Uh-huh.
we can avoid it?
Yeah, I think a lot of people are working on more just like interpretable methods. That actually is a really hard problem, I think, because if let's say you make a statistical model, it's just really hard to know like which of these variables actually is the one that's important that the model considered. So there are people working on this like more interpretable methods. It's like a algorithm like theoretical. It's also a theoretical pursuit. Just Google like AI interpretability, and you'll find a lot of things. And it has to be very yeah. It's just like very domain specific. Like the one that you use in the hospital will be very different from the one that you use at a bank and so on.
Yes, thank you. Thank you, Dr. Don.
I guess in the case of like general AI tools like ChatGPT, you have to drive the ChatGPT as an example. Like you have to make it do what you want exactly
Mm-hmm.
to make it more specific, you know?
Yeah, you do. I mean, ChatGPT is not like it's just a general like assistant, right? Like he only knows what you tell you. It only knows what you tell it. It doesn't know how to read your mind. Although I would say a lot of people are using it as a therapist. I think it's a pretty interesting. There's some articles about this too.
Yes, they are.
Sometimes not necessarily positive. Like I think I see some articles like this. Um, yeah, I think the regulation of AI or just AI in society is very interesting.
They are even people using it like for asking general question like, what do I do today at school, or what do I eat today? And they are getting recommendation from ChatGPT to drive their lives.
Yeah, I mean, you can do that. I mean, I do that sometimes, right?
It's
Like if
fun,
I
right? It's
need to
fun.
write an email, if I need to talk to someone, I'll be like, hey, can you tell me what I should do? I don't know how to do. I don't know what to do. I don't know how
Yeah.
to write this email.
Yeah, sometimes you don't have someone to talk to, so you have ChatGPT now.
Yeah. But I think for the sake of, I think ChatGPT really helps me a lot. But I think for the sake of
Yeah.
learning things, it also makes me feel like I turn off my brain a lot more these days. I feel I'm getting dumber for sure. Yeah, Hala, your point is very, very right. Because this is the part where only the domain expert will know. Because I think what happens is ChatGPT generates a lot of things that is very vague and sounds like it's correct, especially for things like biology or math or just like sciences. But then it's actually wrong. And you wouldn't know it unless you are a domain expert who knows your stuff. So I would also be, have a healthy dose of skepticism when you read some of these things. Does anyone want to say anything else? Or just like thoughts, comments?
I have a questions, Dr. Don.
Okay.
In your opinion, can AI replace a human, especially in terms of disease diagnosis?
Um, I think I would say that AI is pretty good. I think you still probably need humans in the loop to help because it can maybe propose a bunch of suggestions, but it probably cannot replace the entirety of the disease diagnosis. And I will also say, like, in terms of medicine and being a doctor, right? Like, the doctor, the most important part of being a doctor is not necessarily the disease diagnosis part, it is actually the human part of it. Like you go to the doctor because you want to complain about your life. You need a therapist. I think that's actually what most doctors are very good at. They're very good at listening to people and, figuring out like how do you fix not just their disease, but also like, like help them with their psychology and help them to be happier and a better person overall. It's like healing is not just about the disease, it's about like a full body process, right? So I think everyone still probably still need the human contact part of it, and that will probably never change. But I would say that AI is definitely very good at helping people to diagnose diseases these days. Like, it has just like read way more papers than any single doctor might have been able to. And then to Arwa's question on clinical trials, yeah, I think we probably can't skip clinical trials. But then there are also different ways of doing it such that it can become much more efficient. Yeah. Like, for example, finding the right people to do the clinical trials with, like, maybe then you don't have to recruit so many people. Because I would say right now, clinical trial is a very big part of, like, just like the why drugs are so expensive today is because clinical trials are very expensive. So that if a drug company put a lot of money into running a clinical trial, then they will also need to get back the money somehow, right? And then they can only do it by, increasing the price of the drug. So if you can run shorter clinical trials that are just better and more efficient, let's say using AI, but also just a lot of other ways, economic ways too, then you can just have better drugs more quickly and cheaper. So when modeling biological systems, how do we know if a model is accurate or needs improvement? Yeah, I think this is the part that is like kind of hard because I think this is the part that just requires domain expertise. Yeah. I think it just takes a lot of expertise to know and see whether what you're modeling is correct. So you have to kind of work together with both software engineers and computational biologists and these like ML people together with the domain experts like the biologists, because there are just things that the software people obviously are very good at building models, but at the same time the domain experts need to give their feedback on what is the direction to do. I think actually this is like a very kind of a big area, and which is why I think that all of you must be biologists, so it actually really helps even if you don't really like coding or think that, oh, computer science is not for me. You can definitely spend some time learning just a little bit about computational biology, like learning how to code. It definitely be very helpful for you. Like actually for me, I actually learned how to code by self-studying it. I was never a computer science major, but then I just read a lot of books, and I did a lot of questions online. That's how I learned my CS skills. And I think you can also do that too. It just takes a lot of, yeah, it just takes a lot of, like self motivation and drive. And I'm pretty sure all of you are very smart and you can learn how to do this as well. And it will really be very helpful for your future. Okay, kind of gotta go. Thank you so much for the class. And you can email me if you have any questions. I think Rana will also share my email and some of the slides from last week as well.